In this video, we'll be talking about DNA replication. So in the last video, we had discussed about the DNA and the RNA structure, wherein we discussed about the double-stranded structure of DNA. We also talked about how the backbone of DNA was formed by sugar molecules attached by the phosphate bonds. Now, before we start the replication process, we need to just look into few important things about the structure that is important in understanding the replication process. So in our last class, we saw that the DNA strands, or suppose this is the DNA strands, they have an anti-parallel arrangement. That means if one strand is 3' to 5' in direction, this strand will be just the opposite direction. So if this strand is this way, the other strand will be in this direction. This is called as anti-parallel arrangement of the DNA strands. So these are the anti-parallel strands. Now, why we have this 3' or 5'? What does it mean? I have already discussed this in the phosphate bonding between the sugar molecules and when we talked about the DNA structure. So basically, the phosphate bond, it connects the 5th carbon of sugar to the 3rd carbon of the next sugar molecules, and that's what gives it a direction. So that is about the anti-parallel structure. One more important thing is whenever a new strand of DNA forms, it always forms only in one direction, that is from 5' to 3'. So this is something we need to keep in mind before we understand the replication process. So let's start understanding the replication process. So let's start with the DNA strand. This is the DNA strand, and these are the bases of one strand, and these are the bases of the other strand. Before we start building the daughter strands, we need to separate these parent strands. So for the separation, we need an enzyme that is called as helicase. Helicase is the one that breaks the helices. So it is helicase. So what it does is it's kind of an unzipper molecule, unzipper enzyme, which cleaves the DNA strands and it opens it up. So after the zipper molecule works, what we have is something like this. So the zipper molecule has opened up a part of the DNA molecule, and we have the left side still attached and the right side is opened up. So because of the opening of the DNA molecule, now we have these bases which are available for us so that the complementary nucleotides can come and bind to them, forming the new DNA molecules. So it's like each strand acts like a template. So let's just call it, this is a template for the formation of a new strand. Meanwhile, the helicase will continue the cleavage over here, and we already have the strands open up here. So before we progress, let's label, since we already saw that one of the strand is in the direction of 3' to 5', the other strand will be in the direction, just the opposite direction, that is 3' to 5' on this side. Okay, so now we know that DNA can form only in one direction, that is from 5' to 3'. So this strand, which is in the direction of 3' to 5', this is called as parent strand because here it's simple. Here the DNA can form in this direction because it can form from 5' to 3'. So as helicase continues to cleave the DNA, the parent DNA molecule, a daughter strand can form here in this direction. There's no problem. So this is called as the leading strand. This daughter strand which is forming here is called as the leading strand. This is the leading strand, and it's formed continuously. As the helicase continues to open up, this strand continues forming in this direction. Why is it so? Because we already saw there's no problem for DNA to form in the direction of 5' to 3'. The enzyme that is involved in the formation of this leading strand, we'll write it here, it is the DNA polymerase 3 enzyme, and this is again an important point. DNA polymerase 3. Now what happens to the other strand? Now in other strand we have a problem. If you want to form the daughter strand in this direction, that's not possible because that direction will be 3' to 5', and we cannot form a daughter strand in this direction. Now if we want to form the daughter strand starting from 5' to 3', that is in this direction, the problem again here is that we do not have the whole DNA strand open up here. So we start building and then the rest of the strand again opens up, then we have to build, so it's build back the remaining portion again. So it's like this side is built in fragments. So let me explain it. So how it goes is, now we have the part of DNA open around approximately this much, so we can have this much segment built. Now as we progress, we'll have more part open. So I'll be drawing only the lower half now. So we already have this part built up, and our helicase is now reached somewhere here, and we have this extra zone which is now available for building. So let's build that zone up. So this is again the next zone that builds up here. So in this strand wherein we are building in opposite direction, that is we want to build from 3' to 5', but we cannot build that, so we are still building from 5' to 3' direction, but we cannot build whole together because helicase is still opening up the DNA molecule, so we build it in fragments. And this fragments of new daughter strands which are built up this way, they are called as Okazaki fragments. Okazaki fragments. So once all the fragments are built up, later these fragments are joined together, and this is how the other strand, that is also called as the lagging strand, is built. Now let me just talk about a few more details about the lagging strands. Lagging strand which is supposedly built in opposite direction, but we cannot build it in opposite direction. So what we do is we build it in the leading direction but in fragments, which are called as Okazaki fragments. Okazaki fragments. So when we start building the lagging strand, there are few enzymes that come into play. First is Rna Primase. So before one Okazaki fragment is built, there is an enzyme that comes into play that is called as Rna Primase. This enzyme Rna Primase, it kind of builds a small RNA fragment here, and on the top of which the DNA polymerase 3 enzyme builds the part of the DNA. So DNA polymerase builds a Okazaki fragment. So that's how it works. So once all the Okazaki fragments are built, what we will get here are alternate fragments of the DNA, that is Okazaki fragments, and then we get these RNA fragments, and then the alternate DNA, then the RNA. At the end, when the whole strand is ready, then we have one more enzyme that comes into play, that is DNA polymerase 1. Let me write it here, DNA polymerase 1, which comes and replaces this RNA, and it completes the whole DNA fragment of the lagging strand. So this is how the DNA replication takes place. So let's just summarize it once more, wherein we'll be talking about highlighting all the important enzymes in here. So the first thing that we discussed was that the DNA strands are anti-parallel. That is, if one strand is 3' to 5', the other strand will be 5' to 3'. This is called as anti-parallel. The DNA is always built only in the direction of 5' to 3'. So when the DNA replication process starts, the enzyme helicase zips open the DNA molecule. So we have two strands open. The strand that runs from 3' to 5' acts as a leading strand on which the daughter molecule can be built from 5' to 3' direction. So there's no problem. But the strand that runs from 5' to 3' is a problem, because we cannot build from 3' to 5' direction. So we have to build from 5' to 3' direction. But since the zipper molecule or the helicase is still opening up, we need to wait. So we build this in segments which are called as Okazaki fragments. So there is a role of RNA primase which starts the building process. Then the DNA polymerase 3 builds up the fragments, and at the end the RNA part of the fragments, that is in between the fragments, is replaced by DNA polymerase 1 to complete the whole DNA fragment. So this is how the process of DNA replication takes place.